Okay, our next speaker is my co-chair, Hanieh Farsibaf from UT Southwestern.
Thank you so much for having me. It's my pleasure to share my recent paper in Dr. Daniel's lab. So I'm a postdoc in his lab, and in his lab, to understand the mechanism of what drives cancer cells, we develop also a computational framework to quantify 3D images. What you observe here is that the melanoma cancer cells that we detected using the light sheet microscopy, but there are a few tools that we can quantify these 3D images in with high resolution. So here we visualize the NRAS localization for on the melanoma cancer cells that has the NRAS mutation. The way that the NRAS organized at the cell membrane is critical for the survival signal. So from decades of research, we learn in cell biology how the membrane protein is organized at the cell membrane is really critical for cell functions. For example, if they are uniformly distributed versus they're highly polarized. So to quantify this spatial scale in 3D, it's really challenging. And also because of the cell geometry variation, even within a single experimental condition, as I show here, it's melanoma cancer cells in untreated cells. We visualize the NRAS localization, but as you will see, to understand the similarity of this organization, it's really hard computationally. So for this talk, I want to introduce a framework that how we learn from a 3D image, how we learn the biology from the 3D image through the cell signaling organization. So the framework has 3 main modules that we create a molecular pattern from a 3D image, and then we analyze this pattern, and then we try to understand the biological insight. So I just give one example here, but you hopefully at the end of my session, you feel how easy you can implement it for your data. So we collected data using the light sheet microscopy. Here is the NRAS localization in melanoma cancer cells. We have a tools to segment our cells and create a triangle mesh for further analysis. And we can map the fluorescence signal from raw image to this triangle mesh. So this is the first module of our framework. The second one is how we can parameterize this pattern in 3D. Idea is similar to Fourier transform. I hope most of you are familiar with the Fourier transform, but just a brief introduction that in Fourier transform, we have a series of sine and cosine function from low frequency to higher frequency. And when we have a signal, we ask the question, which of these sine function is important for this signal? As it's kind of obvious, you can guess that, for example, the first frequency, because we have one big peak and valley, is important. And also we have the frequency level 10 is important to describe this local fluctuation. So since we have a new signal, we have the same sine cosine function, but the coefficient for each function is different. So we use this idea for a 3D analysis. So when we have a shape of the cell, for example, it's again the same melanoma cells, when we have two patterns, we can use the Laplacian operator to create the eigenfunctions like this one. It's like as a Fourier transform from low frequency to higher frequency level. And we can also obtain the eigenvalue, which descriptive for the shape, and ask the question, if we have a given pattern on this 3D shape, which of this eigenfunction is important? And create the profile of this coefficient, which I call it the eigenprojection. And this eigenfunction is unique for this shape. For a new shape, we need to create a new basis. So we call it the cellular harmonics to describe our pattern. So this is the second module of our framework. So we have a 3D raw image, and we can parameterize our pattern for this 3D raw image. The next question, how we can understand the biology from this framework. So as you also see here, the eigenprojection profile is hardly to explain. So it's highly fluctuated. Then the way that we try to simplify that using the quantum mechanics. So you are familiar probably with in chemical quantum, you are familiar with the orbit and the energy spectrum. So the way that we describe it, instead of asking which eigenfunction is important, we ask which special frequency is important. So we kind of collapse all the eigenfunction within one frequency level and then create this energy profile. So since we can parameterize our signal, we can reconstruct it. Is it similar to a Fourier transform again? So by adding more and more of this eigenfunction, we can create our original pattern. As you will see here, for example, in this case, we need up to 62 levels. And since we have our parameter, we can filter out the signal in a precise way. So we can consider the low frequency versus the high frequency, where biologically the low frequency gives us the global behavior of the pattern, while the high frequency gives us the insight about nano scale fluctuation of the protein aggregation. So this is the whole package of the whole picture of our package, and you can find it on the GitHub. It's written in MATLAB. It's really step by step. So, and we provided 3 examples. Hopefully you can follow the instruction, and if there is any question, comments, I would be happy to take by email. So, but this is not just the framework that we have has to always be validated. So the way that we validate our framework, we took one observed cell geometry and we painted them by introducing the polka dot pattern to show that if we have just giant single aggregation, we need only low frequency level. But when we have more and more number of this protein aggregation, we need higher frequency level to describe. And the beauty of this framework is in shape invariant. Regardless of the shape, similar pattern has a similar profile. And we tested our framework for 67 cells. As you see here, there are highly variable geometry from a blebby cells. We have elongated cells. We have some spiky cells here. I don't know if, oh, you can see my cursor. And we have these dendrite cells that they have a different protrusion. But as I said, similar pattern have a similar profile. So I just wanted to show you as a demo in this talk, but we can have more application in the paper that we recently published in the Nature Computational Science. So as I said, as a kind of a summary here and more application, that we can quantify the different spatial scale of the protein organization across a cell population with diverse geometry. We can use it for quantifying the similarity of our organization for a single experimental condition across different cells, because in reality, we don't want to perturb our geometry. So we wanted to be able to compare them, although their geometry are different. And we can quantify the redistribution of our pattern when we have, for example, the untreated cells versus the drug-perturbed cells. So we can identify how this spatial scale of this organization may change. Also, we can find a similarity between our protein aggregation and morphological motifs. For example, in this case, we detected the bleb on these blebby cells, and we tried to show how this blebby cells, how this bleb distribution is contribute to the NRAS localization. And by these slides, I want to thank to my PI, Dr. Dan User, who provide a collaborative environment for us to learn biology, computer science, and microscopy. And also, I would love to thank to my other colleagues in Dan User, Fiolka, and Dean Labs. And thank you so much for listening to my talk, and I would be happy to take any question.
Thank you, Hanieh. Questions?
Beautiful talk. I love this stuff. Along the time direction, so the fourth dimension, if you will, how dynamic are these protein patches, and therefore how this analysis, right, depends on, I guess you don't want too much dynamical changes. Well, you don't capture that the complexity, because it's going to change the next second. I was wondering if you could tell me what the typical time scale is. It's seconds, minutes, hours for such a patch to move around or disappear at least.
So actually, the analysis is for a single time point. So what, right, for example, we had data for PI3 kinase, and we added the PI3 kinase inhibitor. And along with our imaging, we could show that from a polarized distribution, it's changed to a more uniform distribution. So this, for example, this profile is from a low frequency shifted to the higher frequency. So as just to address your question, that if within time 1 and time 2, there is no significant difference in the spatial scale, we cannot capture that one. But if there is a significant change, we can easily capture that.
Thank you. Other questions? I'm wondering, in the first step where you're creating the mesh surface of the cell, is that in a Cartesian space? Or
yes.
Okay.
Yes.
And then you described some features as like spiky and things like that. Can you actually count the number of protrusions and their kind of distinct morphologies? To say like this is a philopodium and this is a bleb. I know you guys analyze a lot of blebs, so I think you can count those, I guess, but can you distinguish bleb from philopodium from lamellipodium and count them?
So actually, one of our former postdoc, who is a professor in Minnesota, Megan, yeah, so she did this package. So she developed a package to detect the morphological motifs, the blebs versus the philopodia versus the lamellipodia. And we also have some ongoing project, Felix, in our lab, that he also developed a pipeline that we can capture this protrusion and classify them. Yes.
Yeah, we've talked to Megan about applying that to some of our data, so that's great.
Oh, yeah.
And then in that paradigm, can you track individual protrusions in their lifetimes and things like that?
I believe we can. I think Megan also worked on that one for the mesh tracking. So we, the way that, as I know, she explained, developed that framework using the similar Laplacian framework to just capture the eigen function within the time series. And we're also using the unwrap framework that we can do the track the protrusion over time. Yes.
Great, thank you. Any other questions? All right, let's thank Hanieh again.